Imagine you're unwell and you need an injection to get better. You trust that what's in that syringe is pure, sterile, and ready to help your body heal. But what if it contained invisible troublemakers? I'm talking about pyrogens. These aren't living bacteria or viruses themselves, but rather, they are substances, often bits of bacterial cell walls, that can trigger a dangerous fever if they get into your bloodstream. They are the unwelcome guests that can turn a healing medicine into a harmful one, causing chills, aches, and a spike in body temperature that your already ill body simply cannot handle. It's a serious risk we absolutely have to eliminate from medical treatments. So, how do we find these invisible culprits? We can't always just look for bacteria because even sterile products, ones with no live germs, can have pyrogens left over from the manufacturing process. This is where biology gives us a brilliant, if rather old-fashioned solution. We need a living system that reacts to pyrogens in the same way a human does. For decades, the answer has been the humble rabbit. Their bodies are remarkably sensitive to the same fever-inducing substances that affect us. By using them as our biological detectors, we can get a clear, reliable signal about the safety of a drug before it ever reaches a patient. The core purpose of the rabbit pyrogen test is, therefore, one of profound public safety. It's a crucial gatekeeper in the world of pharmaceuticals, especially for anything that will be injected directly into the body, like vaccines, intravenous fluids, or antibiotics. These products bypass many of your body's natural defenses, like stomach acid or the skin barrier, going straight into your circulation. This direct route means there's no room for error. A pyrogen-contaminated batch of medicine could have devastating consequences, so every single batch must be rigorously checked to ensure it is free from these fever-causing agents. It is a fundamental step in guaranteeing that medicine helps, not harms. This might sound a bit like something from a bygone era of science, and in many ways it is. But the principle remains incredibly sound. The test isn't just looking for a specific chemical, it's asking a living organism, does this substance cause a fever? The rabbit's physiological response gives us a direct, real-world answer. If the rabbit's temperature stays normal, we can be confident the medicine is safe for a human. If it spikes, alarm bells ring, and that batch of medicine will never see the inside of a hospital or clinic. This simple biological check has saved countless lives by ensuring the purity of our injectable medicines for nearly a century. Not just any rabbit can be a candidate for this important job. The test requires healthy, mature rabbits of either sex, usually of the New Zealand white or a similar albino strain. They must weigh at least 1.5 kilograms and be free from any signs of illness or injury. These animals are our sentinels, so their well-being and baseline health are paramount to getting an accurate result. Before they are ever used in a test, they undergo a quarantine period. This ensures they are not carrying any latent infections that could interfere with the test by causing a fever for reasons unrelated to the sample being tested. They are, in essence, highly valued members of the quality control team. Their care and housing are meticulously controlled. The rabbits live in individual cages in a quiet room with a constant, comfortable temperature, somewhere between 20 and 23 degrees Celsius, and controlled humidity. This stable environment is critical because any stress or discomfort could cause their body temperature to fluctuate, which would completely invalidate the test results. They are fed a standard, balanced diet of rabbit pellets with no sudden changes and have constant access to fresh, clean water. They are also handled gently and regularly by the laboratory staff to get them accustomed to human contact, which helps keep them calm during the procedure. It's all about creating a stress-free, stable life for them. Before a rabbit is approved for official testing, it often goes through a sham test. This involves going through the entire procedure, being gently restrained, having its temperature taken. But instead of the test substance, it is injected with a sterile, pyrogen-free saline solution. This process serves two purposes. Firstly, it acclimatizes the animal to the handling and the procedure, ensuring it doesn't get scared and experience a stress-induced temperature rise. Secondly, it confirms that the rabbit's temperature remains stable under test conditions, proving it is a suitable and reliable subject for future, real tests. Only calm, non-reactive rabbits make the final cut. The health of these animals is continuously monitored. Their weight is checked weekly, and any rabbit showing signs of illness, significant weight loss, or an unusually high or variable temperature is immediately removed from the testing pool. Furthermore, a rabbit that has been used in a test that resulted in a fever cannot be used again for at least 2 weeks. And if it has been given a test sample that was treated to remove pyrogens, it can't be used again at all. These strict rules are in place to protect the welfare of the animals, and just as importantly, to uphold the integrity and reliability of this vital safety test. The success of the rabbit pyrogen test hinges on meticulous preparation, where every piece of equipment and every drop of solution must be pristine. The primary rule is that everything that comes into contact with the test solution must be sterile and, crucially, pyrogen-free. This includes the syringes, needles, and all the glassware, like beakers and flasks. These items are typically made pyrogen-free by heating them in a special oven at a very high temperature, often around 250 degrees Celsius, for a prolonged period. This process, known as dry heat sterilization, effectively destroys any lurking pyrogen molecules, ensuring that we are testing the drug, not our equipment. The thermometer used is another critical piece of equipment. It must be incredibly precise, capable of measuring temperature changes as small as 0.1 degrees Celsius. In the past, clinical mercury thermometers were used, but today, digital thermistors or electrical thermometers are standard. These devices are sensitive and provide quick, accurate readings. The thermometer is carefully inserted into the rabbit's rectum to a consistent depth, usually about 7.5 centimeters, to get a stable and reliable core body temperature reading. Consistency is key here. The depth and duration of insertion must be the same for every single measurement to ensure the data is comparable and accurate. Preparing the test solution itself requires the utmost care. The drug or substance to be tested must be dissolved or diluted in a sterile, pyrogen-free saline solution. The final solution must be at room temperature before it is injected, as injecting a cold or warm liquid could itself cause a change in the rabbit's body temperature, creating a false result. The dose to be administered is carefully calculated based on the rabbit's body weight, following the specific guidelines laid out in official pharmacopoeias. Every step, from reconstituting a powdered drug to drawing it into the syringe, is performed under sterile conditions to prevent any accidental contamination. Before the main test begins, a control test is often performed. This involves injecting a known pyrogen-free saline solution into a group of rabbits to confirm that the animals, the environment, and the equipment are all in a perfect state for testing. If any of the rabbits in this control group show a fever, the entire setup must be re-evaluated. It's a final quality check before the actual product is tested. This systematic approach, with its layers of checks and balances, is designed to eliminate any variables, ensuring that if a fever does occur during the main test, it can only be attributed to the presence of pyrogens in the medicinal product. The test day begins with a crucial first step, establishing a baseline. About an hour before the test, the initial temperature of each rabbit is recorded. This is their normal resting body temperature. To be eligible for the test on that day, a rabbit's temperature must be within a specific range, typically between 38.0 and 39.8 degrees Celsius. Any rabbit with a temperature outside this range is excluded, as it might be stressed or unwell. A group of 3 rabbits is used for each test. Within 30 minutes of the initial reading, the test solution is slowly injected into the rabbit's ear vein over a set period. This location is chosen because it's easily accessible and provides direct entry into the bloodstream. Once the injection is complete, the waiting and watching begin. The core of the test is monitoring how the rabbits' bodies respond. Their rectal temperatures are recorded precisely at 1, 2, and 3 hours after the injection. Each rabbit's response is judged individually. We are looking for any rise in temperature compared to its own initial baseline reading. The highest temperature recorded for each rabbit during this 3-hour window is known as its maximum temperature. The difference between this maximum temperature and its initial temperature is the key value we need to determine the outcome of the test. It's a simple yet powerful measurement of the pyrogenic effect. The interpretation of the results is governed by strict, clear rules. The test passes if 2 conditions are met. First, no single rabbit can show a temperature rise of 0.5 degrees Celsius or more. Second, the sum of the temperature rises for all 3 rabbits combined must not exceed 1.15 degrees Celsius. If these criteria are met, the product is declared pyrogen-free and safe for human use. This dual-check system ensures that even a moderate response in several animals, or a stronger response in one, is enough to raise a red flag. It provides a robust safety margin, protecting patients from potential harm. But what if the results fall into a gray area? If the initial test doesn't meet the pass criteria, but also isn't a clear fail, the test must be repeated. This typically happens if the combined temperature rise is more than 1.15 degrees Celsius, but less than 2.65 degrees Celsius. The repeat test uses 5 additional rabbits. The data from all 8 rabbits, the initial 3 plus the new 5, are then combined. The product ultimately fails if more than 3 of the 8 rabbits show an individual temperature rise of 0.5 degrees Celsius or more, or if the sum of the rises for all 8 exceeds 3.3 degrees Celsius. A fail means the batch is contaminated and must be destroyed. This rigorous multi-stage process ensures that only the safest products make their way to patients.